And it says I'm recording. So my last step, Jessica, is to go to the PowerPoint presentation on the chemistry of life.
Okay.
Can you see the chemistry of life slide and maybe see me and hear me too?
I just see the slide.
Okay, no headshot of me. That's probably better for you. But you can see the chemistry of life, right?
Oh, I can see you now.
Okay, good. Well, hopefully everyone is on the same page. So let's just talk about chemistry. It's that scary word. You take biology, which is the study of life, and you're thinking, oh, I'm going to go study animals. But you end up a lot talking about molecules, about cells, about atoms. And people are often confused. Why do we spend so much time talking about these issues? And the reason is that's where the answers are. So many of the problems we have, or the situations we'd like to solve, or the things we'd like to develop, are answered at the chemical or cellular level. So this chapter is a review chapter that we're going to do in a very structured way. We're going to define the properties of living cells and of life. We are going to talk about pH, cell potentials, exchanges with the environment, and much more. These subunits that we call atoms, made of protons, neutrons, and electrons. So remember that first. There are 3 subunits, and at the atomic level, they make up all the atoms in all the universe. Just 3, protons, neutrons, and electrons. We're going to talk about chemical reactions, how atoms are held together, and how atoms bind with each other to produce molecules, starting with small, simple molecules like water. When I say a small molecule, I mean a molecule that has a definite formula. H2O, that's always water. And in living molecules in our body, there are some molecules that have definite structures, like glucose. There's always C6H12O6. It's always the same thing. But glucose forms a monomer, and you can take those glucose like a brick and build other things out of them. So the monomer is glucose. When you string glucose together, you get polymers. And this is how you get fibers and nets and rings, is from building these monomers into large macromolecules. And they grade right into the organelles in our cells that make up our living cells. The cells make up tissues, the tissues make up organs, and so forth. So chemical reactions are always going to inform us about how products are formed from reactants. Through the bonding of atoms, we're going to see that atoms don't have a fixed form. They vary with respect to the balance between their protons and electrons, ions, and the number of neutrons they carry in the nucleus, isotopes. And these affect the metabolism of cells. Here's our standard picture of an atom. It's a terrible picture. What is it good for? It's good to show you that the protons and neutrons are in the center, in a place they call the nucleus, and the electrons orbit around them. An orbit just like the orbit of the Earth around the sun. That electron shell has a specific energy level, and it has a specific shape. But these 3 drawings represent our first element, or atom, the atom that we call hydrogen. The left form is the balanced form because there's 1 proton and 1 electron, 1 plus, 1 minus. Its net charge is 0. And this is what we call hydrogen. But hydrogen in this form is fairly unstable. It turns out that we do see forms with neutrons in the nucleus. The middle is called deuterium. Notice how it's the same as hydrogen 1, but hydrogen 2 has a neutron because that neutron raises its mass to 2. That's called hydrogen 2. On the far right, we see hydrogen 3, or tritium. This has 2 neutrons in the nucleus and a mass number of 3. So Here's the first thing to recognize. These are all hydrogen forms because they have one proton in the nucleus. That's called the atomic number, and that identifies the element always. If the nucleus has 6 protons, it will be a form of carbon, always. These forms that have different neutron numbers are called isotopes. Now we're going to see one more form, and I'll relate this to hydrogen. The stable form of hydrogen 1 is when the electron is given away and the proton exists free. Because there is no electron orbiting it, it has a plus charge. An unequal number of protons and electrons gives you a plus charge if there's more protons, or a negative charge if there's more electrons. This is called an ion. So atoms can form ions if the number of electrons and protons are not equal. They can form isotopes if the number of neutrons vary in the nucleus. And that begins our approach to the periodic table. Let's try to bring this home and talk about living things. Living things are made not of the 118 elements that we currently have in the periodic table, but as all cells, and by the way, this is not just human beings. If you go back to biology lab and you look at those amoeba and parameciums swimming around in the pond water, this table is true for those as well. 4 elements make up 95% of the total body weight of a cell or a living organism. And those first 4 elements are oxygen, carbon, hydrogen, and nitrogen. 95% of our weight. Now, a good part of that is because we're all based on water. So since we have so much H2O, we have, as you can see here, 82% of our weight could be just water. Carbon is really the central element of living things because each carbon atom can form 4 strong covalent bonds. That means it can bind into long chains or rings or form nets or layers from the extensive ability to accept bonds with side groups and to form complex polymers. Now, in the human body, we have a lot of calcium only because we have bony skeletons. Since we're vertebrates, we have a lot of calcium, almost 2% of our body weight, but most of that is immobile as a hard crystalline rocky-like solid that makes up our bones. Then we have phosphorus, and you notice we're down to an element that only is 1%, but it's very important in bones and teeth. But it also forms a critical part of 2 very important kinds of molecules, proteins and nucleic acids. Now we're down to potassium and only 0.4%, and we're picking up ions and elements that are present in very low concentration, starting here with 2/10 and going down to 2/10,000 of the first 12 or 13 elements in our abundance chart. So you can see these traces, by the way, potassium on the previous chart, sodium, chlorine, magnesium, sulfur, they may be present in small amounts, but those small amounts are critical, meaning that if we have a magnesium deficiency or a sulfur deficiency, we have a serious metabolic problem from the absence, that deficiency disease. We're all familiar with iron in many forms in the world around us. In our body, its main form is as the central ion in hemoglobin, which allows red blood cells to carry our oxygen. But again, only 7/1000ths of our body weight. Atoms can form bonds that are strong in 2 ways, by transferring electrons or by sharing them. This shows the first type where an ion is transferred. Those rings around the labeled symbol for the atom, Na is sodium, and that purple disk represents the nucleus of the sodium atom. And you'll notice there are 3 rings representing different energy levels. These are actually called the 1s, 2s, and 2p orbitals. The thing about orbitals is when they're full, they're stable. When they are not full, they react with other atoms in different ways. But if you have an outer ring like sodium where it can hold 8 electrons, but you can see there's only 1, this sodium ion really is more stable if it gives away that ion, that electron. Now the chloride, you'll notice that 3rd ring has 7 electrons, so it attracts electrons very strongly. It's called electronegativity. So when you put sodium and chloride together, an electron is transferred, stabilizing both atoms and causing them to bond together. Let me see if I can get a pointer going here that you can see. Now this is a sodium chloride molecule, and this bond is an ionic bond. Ionic bonds transfer the electron and hold these two together very, very tightly. Now ionic bonds are the basis of those things that dissolve well in water. These are called very polar molecules, very soluble, and you are familiar with this. Drop a teaspoon of salt in some water and sit and watch, and it simply disappears. Because water is a polar solvent, it dissolves these ionic molecules into their plus and minus ions. So the water gets salty, and other ionic substances that are dissolved in that water give the fluid the ability to conduct electricity. So we call these substances that dissolve electrolytes. You've probably known someone who had a flu or a diarrhea with vomiting, and that caused them to become very ill and require hospitalization for electrolyte replacement. It's these kinds of electrical conducting substances, the ionic bond substances, that have been depleted. Now this is a list of our electrolytes. You'll notice sodium chloride, potassium chloride, calcium, phosphate. These are all highly charged ions that dissociate and dissolve easily in water. If it is a molecule that will form an ionic component as dissolved in water, the ions released in water, we call that an electrolyte. And these are the ionic or hydrophilic, water-loving substances in our world. Now, the second type of bonding comes from sharing electrons. And you can illustrate that most easily with hydrogen gas here. As I said, the hydrogen atom alone is not very stable. It will exist as hydrogen ion in solution because of the polar nature of water. But you can bond 2 hydrogen atoms together and fill this first ring, the 1s orbital. It only holds 2 electrons. So you'll notice the electrons are between the 2 nuclei, in this case only a single proton. That means that each of these can feel the influence of 2 electrons. And that is a lower energy state and consequently a more stable molecular configuration for hydrogen. This sharing of electrons, completing the outer shell, is called covalent bonding, sharing. The electrons are between the nuclei and exerting their charge influence on both of the nuclei. Oxygen is the same thing because this outer ring, you'll notice the light yellow, there are 6 of them, and the darker orange, there are 6. So there are 2 spaces for more electrons in these outer rings. But if you share 2 electrons from each atom, then these outer rings each act as if they have 8 electrons, and oxygen gas is a stable molecule from covalent bonding. Here in these inorganic molecules are 2 other interesting molecules. This is carbon dioxide, which forms not 1 CO bond, but 2. The central carbon now feels the influence of 8 electrons because it's sharing 2 with this oxygen on the right and 2 with this oxygen on the left. That stabilizes the CO2 molecule by lowering its Gibbs free energy. Nitric oxide, you'll notice, goes a step further, forming, because of the outer shell of nitric oxide, completing it with 8, sharing 4 electrons between the nuclei. So where there are 4 electrons here, you will form what's called a double bond. Each pair forms a single bond. You can see that over here. Oxygen is double bonded together. Carbon dioxide has 2 double bonds, and nitric oxide a single double bond. But it does illustrate the characteristics of these atoms. Oxygen wants to form 2 bonds because it needs 2 electrons. Carbon wants to form 4 because it needs 4 electrons to complete its outer shell. This is what happens when you have a water solution, and we've dropped in sodium chloride molecules. But the water molecule has this asymmetrical shape. You see the central oxygen, which is huge, compared to the small hydrogens that are bonded to it. Those hydrogens form a definite angle. It's not linear or symmetrical. And the pull from the oxygen nucleus is stronger than the pull from the hydrogen nuclei. So that means the electrons in the cloud spend more time up at this end, giving it a slight minus charge. Because there's electrons, more of the time is spent up at this end of the molecule, there's a relative scarcity here, and that gives a slight positive charge to this hydrogen end. So this molecule, even though it's neutral, same number of electrons and protons, the asymmetry of their distribution gives a slight positive and slight negative charge, just like a battery or just like a magnet. Now that means when you drop the sodium chloride in, that big negative chloride, because it has this minus charge, causes these molecules to spin around and attract these positive ends. So bam, 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 the water molecules come pasting in against this molecule, and they literally kind of work their way between the sodium chloride and free the chloride ion. Likewise, the negative end of the oxygen end of the water molecule, bam, it's hitting the positive charged sodium. So it's surrounding these ions with what are called hydration spheres. This is why salt disappears spontaneously when you drop it in water. And it's why it stays in solution until you evaporate the water away. When you evaporate the water away, the sodium binds with the chloride. In the absence of water, you get that same salt crystal back. You also know that sugar, glucose, or our table sugar, sucrose, does the same thing. Although strongly covalently bonded, the water molecules can pry these individual glucose molecules out of their polymers, out of their crystalline forms, and form hydration spheres that liberate them in solution. Now, properties of water are special because of this polar molecule. It divides the world into hydrophilic and hydrophobic compounds. Hydrophilic, like electrolytes, those things that dissolve. But hydrophobic, those things that avoid water, will not mix with it. You know that oil and water do not mix. Oil, wax, waxes, oils, membranes, all of the fats are all hydrophobic compounds because they lack the polar nature. Water has a pH property because of the presence of hydrogen ion, and because pH is a great reactive characteristic of water solutions. In chemistry, how often did you do the reaction by taking the solution and adding acid? That hastened the reaction and the production of product. The same thing is going on in our body with pH control. Electrolytes create the water that is the solvent of life, the carrier for all of our metabolism, the chemical reactions of our body. But also water can be either a reactant or a product. It can actually be consumed in a reaction, or it can be generated by the chemical reaction as a product. It leads to a number of properties of water. Liquid water carries a lot of heat, so it remains liquid over a wide range of temperatures. It produces a thing called surface tension that acts sort of like the top of a trampoline. If you've ever watched a water strider dance across the surface of a creek, its leg is actually coming out and forming a kind of a scoop-like foot with hairs, and it distributes its very limited weight on the surface of the water and actually presses it down like a trampoline. The surface tension of the water, the cohesion among the water molecules attracting one another, creates a surface that it can actually stand and walk on. Water expands when it freezes, and it also moves through tiny tubes by sticking to the walls. Water is going to be the basis of our metabolism. Now, there's a lot of liquid water that's outside of living systems. The pH is described, the powers of hydrogen is what that stands for. The concentration of hydrogen ion is based on the fact that pure water spontaneously breaks down to hydrogen and hydroxide at the concentration of 10 to the minus 7th. So pH 7 is pure water. If you have additions to that water so that the solution maintains a higher concentration of hydrogen ion, then that pH the hydrogen ion concentration is going up, so the concentration will go from 10 to the minus 7th to 10 to the minus 5th. That would be pH 5. The lower pH indicates a higher hydrogen ion concentration. If you add substances that consume the hydrogen ion and enhance the hydroxide ion, then the pH goes up above 7. If the hydrogen ion drops to 10 to the minus 9th, that's pH 9. And now we're in a basic or alkaline condition. The main pH control in the body of the human is controlled by the blood plasma, which ranges here, 735 to 745. So think of 7.4, slightly alkaline, just like seawater. Now, this slightly basic pH maintains most of the pH conditions throughout the body. And in general, I want to tell you that from place to place in the body, there are places where the pH changes from this 7.4. The production of CO2 changes the pH. The manufacture of hydrochloric acid in the stomach makes that environment pH 2. We can tolerate or adapt to acidic conditions. Basic conditions, if you move from pH 7 to pH 8 inside the body, you are killing cells. If the blood plasma gets off of this 7.45 and moves towards 7.8 or 7.9, convulsions and death will follow. So basic solutions are very harmful to living cells, and you can show this easily in the lab. If you're a microbiologist who can generate lots of Petri plates with living cells, prokaryotes or eukaryotes grown in culture, you can basically test different solutions of pH and find out that the basic ones are the ones that kill cells readily. And here is what we mean. Here's neutral, water, and here's where blood usually is, around 7.4. The ocean water generally ranges from the high 7s into the mid 8s. Kind of similarity there. If we go down the scale, so hydrogen ions going up, we have saliva and milk in the mid 6s. But anything that tastes sour, like tomatoes or grapes, things that we think are tart, or down here at pH 3, beer, vinegar. Vinegar is acetic acid. Wine contains acid. Pickles, which are preserved with vinegar and salt, pH 3. Soda pop is right down here, pH 2 to 3. And we readily eat these as foodstuffs, which means we can handle acids. Stomach acid may get the pH of the stomach down to 1. And that stomach is primarily to protect us against pathogens. But if we go the other way, look at this. If we move 3 units up here to household bleach, bleach is difficult. And even our armored epithelial tissues, our epidermis, which repels water, are harmed if we put our hands in bleach. Here, ammonia, just smelling ammonia causes you to move it out of reach. And over here, oven cleaner, a very, very strong base because it can dissolve these organic compounds. The glaze that forms on your oven is readily removed by strong bases. Now, we've kind of covered the atoms, ions, and isotopes, the basics about water chemistry and electrolytes. What's next? What's next is to recognize that this central atom, carbon, is going to make up all of our organic molecules on a monomer, meaning a fixed formula, and polymer, meaning an indefinite combination of monomers basis. So let's build this in a specific way. I'm going to go down this list by talking about first carbon. Carbon is the central molecule because it can form so many bonds. There's not, by weight, there's not that much carbon in our body, but it's forming the backbone of all organic molecules because, here it is, carbon can form 4 strong covalent bonds per atom. So here's a nucleus of a carbon, and it's bonded to a carbon on one side and a carbon on the other side, but it still has 2 bonds. You can put on a hydroxide and make it an alcohol, or a COOH and make it a carboxylic acid, or an NH2 and make it an amine. Or you can bind it to another carbon up and down and start that carbon as the central point where 2 bonds cross at right angles, beginning to form a net. Now, if you take that carbon, the basis, the linchpin for organic molecules, and you add just hydrogen, what you get is a monomer called a fatty acid. Those fatty acids are very symmetrical, very hydrophobic, and therefore this is the first and simplest of our polymers. There's going to be 4 of them that form the lipids. Lipids are fats, oils, and waxes, and you know, on our level, those things don't mix. Water and oil, water and wax do not mix. Now, that is a statement you're familiar with, but what's its real meaning on the cellular level or on the polymer level? Its meaning is that if you're going to form a structure like a cell, we know all living things are made of cells, you have to form a cell to have life. To have life, you have to have a barrier that separates the outside, the environment, from the living entity, the inside of the cell. And that is performed by the membrane. The membrane barrier cannot be a hydrophilic substance or an electrolyte. It would just dissolve. Hydrophobic barriers form the outer boundary of the cell. Not just the cell, but since humans have membrane-bound organelles, the nucleus, the endoplasmic reticulum, the Golgi apparatus, the mitochondrion, in plants, the chloroplast, and other bound organelles, all have their special region of activity because of lipids. Now we have carbon and hydrogen together, and we're going to add oxygen, and we get a new polymer called a carbohydrate. Now the monomer has a definite structure. Glucose is one. Maltose, lactose are two others that form monosaccharides, single sugars. Now take these monosaccharides and link them into chains or nets. They form fibers. They form energy storage, the things we call sugars or starches. In our body, starches in plants, glycogen, and we nourish ourselves with starches in our diet, but we store glucose as glycogen. Plants also use glucose to make cellulose, which is the indigestible fiber in our diet. There's an irony, is we're eating all of this fiber, and it's made of pure glucose, and it goes through our digestive system, but it's bound so tightly we can't take it apart. So that glucose, the cellulose, the fiber content, comes out the other end as the main substance, the main structure of our feces. Carbohydrates are named for this ratio, CH2O. Carbon, hydrogen, and oxygen in the ratio of 1 carbon to 2 hydrogen to 1 oxygen, or carbohydrate, carbon water. Now let's add nitrogen, and this is going to form a monomer called amino acids. Amino acids are a definite structure, which I'll show you in a minute, and there are an innumerable number you can make in the lab, but all living things from bacteria to blue whales only use 20 amino acids to form the polymers that we call proteins. We'll get to that in just a second. Add phosphorus, and this phosphorus now produces a monomer called a nucleotide. There are only 5 of them in all living things, whether you're a bacteria, a human, or a blue whale. You use the same nucleotides to form your DNA and RNA, and those are called nucleic acids. So what we've tried to do so far is simplify things. All matter on one level, the subatomic level, is made of 3 things: protons, neutrons, and electrons in different combinations. When we looked at a living tissue in a human body or any cellular structure, we find that carbon, hydrogen, oxygen, and nitrogen make up the majority of the biomass, certainly over 95% of the weight of any living thing. When we look at the molecular level, these 4 classes of carbon molecules, lipids, carbohydrates, proteins, and nucleotides, make up over 95% of the biomass of all living things. And so they form a basic structure that's common in living things and important to humans. Here's a picture of a lipid. How symmetrical is this? The black carbon bonded to itself, and each one carrying 2 hydrogens. This upper one, all the bonds are single bonds, and this is called a saturated fat. This is the most hydrophobic form. When you form a double bond, you'll notice you kick off 2 of the hydrogens. See that double bond right there? And it produces a kink in the lipid molecule. It makes it a little less hydrophobic, but it also gives it a place where you can attack it and break it down. This is called an unsaturated fat. So this drawing, because there's 1 double bond, would be a monounsaturated fat. If you have many of the double bonds along the length, it would be a polyunsaturated fat. And you may be familiar with this terminology from dietary concerns. Here is the way we process these long carbon-hydrogen chains. See, saturated, saturated, and unsaturated. These are very hydrophobic entities. They're going to be separated into droplets in our blood. So we bind them to a glycerol, a very hydrophilic head. And this is one of those places where water is a reactant. Notice that when we bind them, going from top to bottom, we call that dehydration, because we form a bond between the carbon and oxygen in each case, and we kick out a water. This is metabolic water. We didn't drink this, we made it. When it's time to utilize this form, which by the way is called the triglyceride form, of our circulating lipids. This is the basis of our energy from fatty acids and fats. When we get there, we're going to break this back down, and we're going to put each fatty acid chain into a mitochondrion to break it down. This is called hydrolysis, and you'll notice it consumes water as we form that bond. So lipids have small structures as well. Here's cholesterol. You've heard that term. Cholesterol, as you can see, is the building block for our sex hormones. Estrogen and testosterone are both built from this starting monomer called cholesterol. We're concerned with cholesterol because of dietary connections to things like heart disease. But we also know that cholesterol is essential for forming membranes, but also for forming our sex hormones, estrogen and testosterone, shown here. Here is a very important lipid, a phospholipid, two long fatty acid chains hooked to a phosphate group, very, very polar. So this strange molecule is sort of like a comedy tragedy mask, a very, very hydrophilic end and a very, very hydrophobic end in a single molecule. The phospholipid is the basic structure for every membrane. It can assemble itself into a bilayer that produces a hydrophobic zone that regulates transport across that sheet from the outside to the inside or the inside out. It is the barrier of life. When you put phospholipids in water by themselves, you'll notice how they assemble here. See these round orange spheres? These are hydrophobic heads. This is a phospholipid because it contains phosphate. This is a glycolipid because these are sugar groups, but they have the long hydrophobic tails. So do you see how the long hydrophobic tails are dominating inside and excluding water? But the outer surface of this micelle is hydrophilic ends of these molecules, the phospholipid and the glycolipid. So water next to this is very stable. So we have a problem with lipids. We can't make them mix, but they have to be stable in water solution. It is the phospholipid and glycolipid that illustrates how this is possible. Carbohydrate. Here are three different drawings of glucose. You can see the space-filling model down here with carbon, hydrogen, and oxygen forming this monomer. Glucose forms the sugar that we define in our dietary language, sucrose, that sweet table sugar, and our body can't actually digest this. We eat sucrose, that's what tastes sweet, and we add water to break it down, hydrolysis, into a glucose and a fructose molecule. Our cells can break down and digest these monomers. So this is a part of our digestion as we process these. The monomers are shown on the right. This structure on the left is called a dimer because it has two subunits, and it is the simplest kind of polymer. If we take glucose and string them together in endless change, we get our polysaccharide, or our polymer for storing glucose in the body. In our body, it's called glycogen. We store that in our muscles and in our liver for quick energy. But there is no limit to how long a chain you can make. You just pile on more glucose if they're available. This is the monomer of proteins, the amino acid, a central carbon, an NH2 amine group, a carboxylic acid group, and an alpha hydrogen. Those four parts on the upper part of this diagram are always the same. The R is variable. Change the R to an H, and you have the amino acid glycine. Change it to a double bond with a ring of 6 and a ring of 5. In other words, you're adding 10 carbons and the associated hydrogens and oxygens around that ring, and you have the amino acid tyrosine. These monomers assemble into linear strands by gene expression when we synthesize proteins. So you have several levels of structure. The first level is called the linear sequence or primary structure. And the sequence of amino acids as they occur cause it to fold up in regions of the chain into regular repeating structures. You can see here's a region of an alpha helix. Here's a region that is like a screen, a pleated sheet. Now, once those local regions are folded, the secondary structure, the remaining groups fold that single chain into a three-dimensional structure, in this case, around a central group called a heme group. This is tertiary or third-level structure, and that's what happens with a single chain. But we are not done with this particular illustration because the active chain is not, the single chain is not the active molecule. In our body, 2 alpha chains, shown here, combine with 2 beta chains, each folded around a heme group to provide the active protein that we call hemoglobin, our oxygen transport. So there are many levels that come from the synthesis of these amino acid polymers. Not all of them are soluble and circulating like hemoglobin. Here's basically a helical structure with 3 strands for keratin or collagen. These are insoluble proteins that form structure in our connective tissue, that strap our joints together or reinforce our bones. Another function of proteins is shown here. This is what happens when you take the bare reactants in our metabolic reactions and put them together in a solution. There is an amount of energy that's required to make those reactants bond together. That's shown by the dotted line. This is called the activation energy. So if you just put in the reactants, this activation energy is high enough that the reactants do not combine. Now in the lab, how many times I mentioned adding acid to change the pH? What about if we add heat? Often we would heat a solution to get the reaction to occur. But our body's limited. We have a constant temperature. We can't heat our body up to 60 degrees C. Our body temperature is 37. So this is where proteins as enzymes come in, because the protein binds a reactant and changes its shape and charge. That lowers the activation energy to this red line, which is a low enough energy that it then spontaneously converts to product. So this shows you something about proteins. Throw that protein in and you get reactant to product 1 to 4. Remove it and this reaction stops. So this way gives us a way with the enzyme to control the reaction, turning it off and on. The final monomer is called a nucleotide, and there's a chain of phosphate, sugar, going up and down one side of the molecule, and each sugar has a nitrogenous base attached. This nitrogenous base, one of 5 in DNA and RNA, has the special property of complementary base pairing. So when you put 2 strands together into the double helix, you'll notice how A is always paired with T and C with G. That forms this linear chain into a double helix that's very stable. In fact, whereas proteins break down very quickly and leave the cells, if you break apart cellular integrity, proteins begin to denature immediately. But if you purify DNA and put it in a test tube, you can put it in a refrigerator with cotton in the top of the test tube, test it every 6 months, and there's no negligible change. It's so stable. Complementary base pairing, A to T and C to G, and the nucleotide base sequence here will explain the genetic code. And the connection is that the genetic code for this region will produce one and only one protein. So this is how our genes are coded and how they are related to the protein that they produce. So ATP is another form of these nucleotides. When adenine and ribose obtain 3 phosphates, these are very high energy bonds, and we use the energy from our dietary substances. We release energy through digestion and stick phosphates onto adenine. Those phosphate groups can then be broken down to yield energy when we're doing something like making vesicles or transporting materials across a membrane or pulling chromosomes apart in mitosis. ATP is the energy exchange in the cell. Chemicals and cells behave in a constant and dynamic way. What do I mean by that? Well, I came out as a little package from my mother, and I grew to my adult size. And if you'd known me from when I was 20, you would notice that a lot of my tissues have aged, but my basic size and shape hasn't changed that much. It might be attractive to think that we grow up to a certain size and stop, which means that that building, that growth or chemical reaction slows down and stops, and that's not true. Our metabolism is constant, and we call this breakdown of our carbon molecules and the resynthesis of our molecules. We are constantly remaking our skeleton and bone, our muscle system and muscles, our tissues that are connective tissues and nervous tissues. And this is called metabolic turnover. It's the renewal and recycling that's going on in your body through that constant mature phase that we call adult life. Now, if we look at turnover times, we make this real. In the liver, the total protein in the liver is turned over in every week. So within a week, all the protein that's in your liver right now will be broken down and replaced by newly synthesized proteins. Enzymes are much more labile, may last only an hour or a few days. The glycogen, which is stored glucose, and the cholesterol is constantly being torn out and renewed. Muscle cells, the total protein in your muscle. You know, if you don't exercise in a dramatic way, it looks from the outside like your muscle mass is pretty consistent, pretty constant. But look at this, the turnover rate, the total protein content, and this is important because actin and myosin, the main contractile fibers, are both proteins. It is turned over every month, every 30 days. All of those actin and myosin molecules are replaced with new ones. And the glycogen, the stored energy in your muscle, every day. In neurons, we have more persistence. So phospholipids in neurons may last 200 days. That's upwards of 6 months. And the cholesterol, 100 days. Neurons last longer because neurons are transmitting signals, but more important than that, they are storing information. So what we call memory and personality and the consistent things about us are stored in our neurons. Fat cells, look at this. If you have a constant body weight, and technically I'm on the over the obese line right now, it looks like my fat is very constant and not changing, but this turnover rate, every 15 to 20 days, every 3 weeks, the triglycerides in our body are torn out and resynthesized from the fatty acids that we obtain in diet. So that is an exit, and I'm hoping that everyone received that recording.